Human society is aging. It is associated with changes in molecular composition within us. However, it is unknown whether the changes in these molecules are drivers or passengers of aging. So, in our quest to identify metabolic networks that underlie age-associated diseases, we performed a metabolomic analysis in a specific nutrient deficiency. This metabolomic analysis showed that one of the most dramatically downregulated molecule in the screen was taurine. Taurine is an atypical molecule and is present in abundant amount in our tissues. It is produced within our bodies primarily through liver and some other organs, and it can be obtained through animal-based diet in nutrition. Taurine is not a new molecule. It was identified more than 200 years ago. In 1950s, pet foods were introduced into the society. This led to an increased incidence of diseases in the pets. The research into this process led to the discovery in 1975 that retinal degeneration or blindness in cats is caused by taurine deficiency. In the present study, we show that taurine abundance declines with age in several species. This led us to investigate whether taurine concentration that declines with aging is a driver or a passenger of aging. To address the role of taurine in aging, we orally fed taurine to middle-aged C57BL6J mice because we knew taurine levels were low in them. Taurine-fed mice of both sexes survived longer. The average lifespan increase in females was about 12% and in males was about 10%. A meaningful anti-aging therapy should not only increase lifespan but also health span.
When we treated aged mice with taurine, it was actually very exciting because we saw that taurine supplemented mice had reduced obesity, increased energy expenditure, and an improved functioning of multiple organs such as bone, muscle, pancreas, and immune system, indicating an overall improvement in the health span.
To find out whether these observations hold true in primates, we next performed a monkey intervention study. This analysis showed that taurine-supplemented monkeys had better functioning of a variety of organ systems, including reduced adiposity, better bone density, better glycemic index, and a better functioning of the immune system.
To understand how taurine affects cellular health, we performed RNA sequencing analysis of taurine deficient cells. These analyses revealed that taurine deficient cells exhibit changes in those pathways which directly regulate aging process.
Through gene network analysis, we observed that the genes expressed in senescent or zombie cells that accumulate in our body as we age have the highest degree of genetic interaction among all affected genes. This for us is a good starting point to perform experiments to confirm the role of these processes in taurine mode of action.
To check the relevance of taurine in human studies, we leveraged results from an existing metabolomics study where taurine levels in blood are available for more than 11,000 participants. There, we found that low levels of taurine associated with poor health outcomes in humans. This included higher abdominal obesity, type 2 diabetes, inflammation, and hypertension.
Next, we investigated on this bicycle ergometer whether a bout of exercise modulates the concentrations of taurine in blood. We find that on average, a bout of graded exercise increases taurine metabolite concentration in blood. And this is interesting to us because this might explain part of the health benefits of exercise and of the anti-aging effects.
Of course, association does not establish causation. But these data are at least consistent with our hypothesis that taurine deficiency is a driver of aging.
The results are very straightforward, although it took us 11 years to do this study. Taurine abundance declines with aging in several species, and a reversal of this decline increases healthy lifespan in mice and worms and health span in monkeys. This identifies taurine deficiency as a driver of aging in these species. Next step of this research is us humans. Taurine has no known toxic effects, although rarely used in the concentrations used here, can be administered orally, and affects all the major hallmarks of aging. Although it is difficult to say at the present time whether taurine supplementation is going to be an anti-aging therapy, Based on our studies in several species and our intervention in monkeys, it is reasonable to test it at least.